Ooh, there we go. Sorry, guys. I forgot to press go live, and then I was just talking to myself for a second. So hi, everyone. It looks like my lighting is really messed up. So let me close this blind. That's better. All right. Well, welcome to our live session. As a reminder, I'm Caroline with Fiveable, and I'm really excited to get to talk to you guys about biochemistry today. And I'll have some information regarding how you can keep up with all of your Fiveable good stuff at the end of today's session. So I'm about to jump into a PowerPoint. Again, the topic tonight is biochemistry. Oftentimes tricky. This depends, I mean, it's obviously a little trickier if you haven't had chemistry before. So it just kind of depends. It's variable depending on whether or not you have a strong chemistry background. So a lot of this might be review. I'm going to try to make it as applicable as possible to biology, obviously, because that's why we're here, specifically AP biology. And I'll give you little tips and tricks of how this oftentimes shows up in exams so that you feel best prepared when it comes down to it. So here we go. I'm going to start sharing my screen, but you do have that link in the document, and it should work. I double check to make sure that everyone with the link has access. But we should be up and running then with my biochemistry PowerPoint. So thing number one, we're going to talk about 4 of the most important things that make up all living things, starting with atoms. We're going to talk about atoms. We're going to talk a little bit about ions. We're going to talk about molecules, and we're going to talk about bonding. Because all of those things, while they may not seem super biological, they make up all living things, so we have to know about them. And it's going to come into play a lot when you're talking about the origin of life on Earth, which is a crazy and nuts topic if you think about it, how we got life out of nothing. But we're also going to talk about this a lot when it gets into cells, and we talk about the cell membrane. So you need to be familiar with like what an ion is, so that when I tell you that ions can't make it through the cell membrane without a transport protein, that makes sense to you. Okay, so up here in the upper left-hand corner, you will see an atom, an artistic representation of an atom because it's not quite what they look like. But atoms obviously make up all living things. We have a periodic table of them, all of our different elements. And the atom of the day, or the element of the day for all of biology, is going to be carbon. Okay, so we'll talk about carbon a lot, and I'll tell you why in a little bit. But carbon is going to be our most important atom. Obviously, atoms make up all living things. They are, sometimes my students get confused, they're not living. Neither are macromolecules or anything like that we'll talk about. Cells are the smallest unit of life, but they make up cells, so super important. In the upper right-hand corner over here, you'll see some ions. Anytime I say ion, you should be thinking positive or negative charged thing. Okay, so you'll see a positive ion and a negative ion in that upper right-hand corner. We're going to talk about ions when we talk about bonds in just a second, but usually that positivity and negativity comes from unequally shared electrons, which you can see orbiting those 2 atoms right there. But anything that has a charge to it, any atom or molecule, the easiest example would be like salt, NaCl, sodium chloride, breaks into sodium, which is a positively charged ion, and chloride, which is a negatively charged ion, and that makes them super, super polar, and we'll talk about what that means in a second. In the bottom left-hand corner down here, you can see this guy would be considered a molecule because we have a bunch of atoms coming together with these bonds to form something bigger than just one single atom, obviously. Again, this is like the most basic of basic reviews, but it's good to get the vocabulary down before I start like spewing lots of things at you. And then finally, in the bottom right-hand corner, you see the difference between ionic and covalent bonds, but I'm going to actually not go into detail right yet because I have a whole slide on that in just a second. Okay, so those are the overall things that you need to be familiar with as far as when we're talking about biochemistry stuff, okay, and more on that as we go through. So bonds, 3 types. Hopefully this is predominantly review for a lot of you, but there's ionic bonds, covalent bonds, and hydrogen bonds, okay? You need to be familiar with all of them just because the type of bonds that molecules make oftentimes kind of contribute to how that molecule behaves. And it's going to help you make more sense of why like DNA acts the way that it does versus a lipid versus all the other things that we'll talk about today. So ionic bonds. Here is a picture of ionic bonds. Crazy enough, they usually form between ions, positively and negatively charged things. So as you can see, we have that sodium ion on the left, which is naturally going to be relatively positive, and we have that chlorine or chloride ion on the right, which is naturally going to be negative, a little bit electron hungry. But when they form that ionic bond at the top, they're actually going to share or kind of transfer that electron so that you can see sodium's really, really happy because now it has 2 electrons in its outer shell, which is, if you know anything about chemistry, having 2 electrons in your outer shell makes you very, very happy. And then chlorine is also very, very happy because it has 8 electrons in its outer shell, which is another magic number. So having 2 and 8 electrons in your outer shells make you a full and complete and happy molecule or atom. And so when they're sharing those, it's like friendship is sharing, sharing is caring. Sodium has its 2 little atoms, chlorine has its 8, but really they're just sharing. Okay, good with that? So with ionic bonds, when these things enter water, which we'll talk about in a second, they're going to actually split back up into their ions. So when you pour salt into water or you go into the ocean, the salt water that you're in actually is full of free-floating positive sodiums and free-floating negative chlorines because they will disassociate in water. And we'll talk about why in a second because I have so many slides to tell you about all of water's amazing, unique properties. Okay, ionic bonding. All right. Next, we have covalent bonds. Covalent bonds are going to be a lot more friendly because they're going to have, instead of the electrons kind of being transferred here, which we can see, these guys are actually just going to share them, and you can see they actually will overlap. So on the left here, these 2 oxygens, you'll see they're overlapping their electron orbitals, and that's how they form their bonds. Okay, so I should have mentioned this when we started talking about bonds at the very beginning, but the bonds are always going to form based on either sharing or transferring of electrons. Okay, so we're going to talk like next week when we talk about how the environment on Earth in the beginning of time was probably relatively reducing. We need to know that that means that there's lots of electrons around, and lots of electrons means lots of bonds can form between things. And that's going to help us make more sense of why we got to, make macromolecules in the beginning of time when Earth was just volcanoes and ash and all sorts of crazy things. Okay, so these covalent bonds, they're going to be sharing. On the left, we have something called a non-polar bond. Those are equally shared. So those are that's like an equal friendship where the give and take is there from both sides. So those oxygens are equally sharing those electrons. You can see they're overlapping orbitals, right? And things that have non-polar bonds, we call non-polar, okay? This kind of stuff will not split up in water. So like another good example of things that have non-polar bonds would be a sugar molecule. Sugar molecules share their electrons very nicely between them. They're carbon, hydrogen, and oxygen molecules. And because of that, that will not break up when it's put into water. So when you're dissolving sugar into water, it actually isn't dissociating into those ions like you saw before. It will just stay as smaller little groupings of sugar molecules. Okay? On the right-hand side, a different type of covalent bond. So we're still sharing electrons here. Okay, but this time it is a polar covalent bond because we are sharing unequally. So this is that unequal friendship where someone is putting in all the work. Maybe someone's a little greedy. In this case, the best example of a polar bond is water. Water is incredibly polar, and things that are like water are also very polar. Okay, so as you can see here, we have an oxygen molecule binding to 2 hydrogens, but the electrons, which are represented by these little black dots here, right, these guys are much, much closer to the oxygen molecule than to the hydrogen molecule. So I'm sure you all know when you write H2O, you don't add a plus or a minus charge to that. It's a neutral molecule, but the oxygen in that water molecule is actually quite negative, and the hydrogens on either side are actually quite positive because the electrons are not being shared equally. So oxygen is the most electronegative molecule. So if you've taken chemistry before, you know it sits all the way in the upper right-hand corner of the periodic table, makes it the most electronegative. Okay, so it is very, very, very electron hungry. So it pulls those electrons closer to it, giving it an extra negative charge and giving those hydrogens less electrons, which innately gives them a more positive charge. And this is going to lead beautifully and wonderfully into our last type of bond, which are hydrogen bonds. So because hydrogen molecules are oftentimes a little bit more positive, they can actually form hydrogen bonds with slightly negative molecules of an atom nearby. So hydrogen bonds, as opposed to ionic or covalent bonds, are between molecules, not within molecules. So I like this picture because it shows you the difference. You can see the covalent bond between the oxygen and the hydrogen atom. That's a polar covalent bond there. But between this slightly positive hydrogen and this electronegative negative oxygen, there's a bond because, well, opposites attract, okay? And that is called a hydrogen bond, okay? It's actually, as far as bonds go, a pretty weak bond, but we're going to see that that hydrogen bond actually allows water molecules to do some pretty amazing things. So while it may not seem like a whole lot, because it's basically just like an attraction between a slightly positive hydrogen and a slightly negative oxygen or fluorine or nitrogen, FON. Hydrogen bonds are FON, F-O-N. Because it's just that attraction, they're going to be weaker than like a covalent or an ionic bond would be. Okay? All right. So those are the bonds we're talking about. And again, they're going to help us determine how molecules act, but they're also important to know because they're just the sharing of electrons. And so we need electrons to form more bonds. All right. So our next little segment, so we've talked about the basics, atoms, molecules, ions, and bonds, all super, super important in biochemistry and chemistry alike. Now we're going to talk about water. There's sometimes entire chapters in textbooks, biology textbooks, on water itself because it's crazy, and obviously we would not be alive without it. It's a very, very important molecule. So I'm going to give you just like the overview of the major properties of water because it allows life to happen, and so you need to be familiar with it. As you can see in this diagram, we have our little Mickey Mouse water molecules. They're binded together by those blue dotted lines, which are the hydrogen bonds, which are highlighted for you. And these little guys that kind of look like a music note, it's technically a delta negative, delta positive. That just shows you that there's a partial charge on these guys, even though the molecule itself is not charged. Okay? All right, 4 properties of water that we are going to talk about. So water is capable of cohesion and adhesion, super, super important principle that I'll talk about in a second. That's property number 1. Also, second property, ice floats. Sounds crazy. You're like, yep, I knew that. Of course ice floats. I've known that since I was, I don't know, 2 years old. But we're going to talk about why that's so crazy and why that's so important that ice floats. We're going to talk about this specific heat capacity of water and how water has a very high specific heat and why that's really important. And then lastly, we're going to talk about how water is a universal solvent and can break things down for us, which is really, really awesome and makes it extra special. So, thing number 1, cohesion and adhesion. Okay, so the diagram on the right shows you the basics of what cohesion and adhesion are. So cohesion is water molecule to water molecule holding on, okay, between those hydrogen bonds, right, which is what you saw in this slide 2 previously. Okay, so water molecules stick together really, really, really well. Second property, adhesion, is water molecules sticking to another surface. Okay, so as you can see here, the spider is able to walk on water because there's actually tension in the water from all of the cohesion and then adhesion. Going along with that, another really, really important thing that cohesion and adhesion show are that that we can get water from the roots of a tree all the way up to the top of a tree. And we have redwoods, right, that are over 100 feet tall, and we can get water all the way from the roots all the way up to the top because that's the only source of water for plants is through their roots. Okay, and that only happens because of cohesion and adhesion. So water molecules are sticking together. Water molecules are sticking to the insides of the xylem of the plant, and they're able to essentially climb up to the top of those trees that are like 100 feet Quick question for you, because I want to show you an example. Can you see me in the upper corner of your screen, or do you just see my PowerPoint? Like, do I exist on your screen at all? Or am I just, oh, you see both, perfect. Because I've been talking with my hands a lot, and I'm like, wow, maybe I'm talking to absolutely no one. So that's good to know. So I'm gonna grab really quick, I'm gonna grab 2 microscope slides and a water dropper, and I will be right back to show you a little example. Okay, I'm back. I actually think I'm going to make myself the whole screen because you definitely want to see this. So I have 2 microscope slides here, right? And I'm going to do like a little balancing act. So obviously we all know that gravity exists. It's a very strong pull that keeps us on Earth. If we were on the moon, which has less gravity, we'd be 2 inches taller. So if you've been trying to get taller, you should think about moving to the moon. Okay, but we know that gravity exists. So if I try to put one side on top of another and balance it on the end, it falls because gravity exists, right? So I'm holding on to this slide, but if this slide is balancing on the end, right, obviously that slide is incapable of staying up, okay? The crazy thing though, and it's just an example of how strong water molecules are, if I take a drop of water, this lovely water, I just put a tiny little drop. So all I did is I put a tiny little drop of water on the side. You can kind of see it rolling around. I do that, and then I stick this slide back on top, okay? And I just kind of wipe the water off a little bit. Wow, gravity-defying. As you can see, obviously these 2 slides are together now. And again, I'm not touching this top side at all. I'm actually sliding around right now. So the only thing that's connecting this top side to me is hydrogen bonds. So there's hydrogen bonds between the water molecules in the glass. There's hydrogen bonds between the water molecules. And all of those bonds between water molecules are the only thing that is connecting this top side to my hand, which is preventing it from falling right now. And as if that wasn't enough, I can actually stack. So I have some poker chips because I use them to describe lots of different things. I can actually stack some poker chips on the end here. Now this is I know. Ooh, we're sliding out. Come back. Right, but I can actually stack, which is going to increase obviously the force of gravity because gravity slide. I put too much water on. But gravity is obviously going to act heavier on heavier things, or act stronger, I should say, not heavier on heavier things. So gravity is pulling down on this even more right now. This is like a party trick. You can do it at home for all your friends. Okay. It's my goal, just show you cool things so that you make more friends. But I can keep stacking, and eventually the hydrogen bonds will start to break, okay? But we have a lot of weight, extra weight right now on the end of this, and it's still holding up just because of water molecules. So it's pretty crazy. Looks like the bonds are starting to break, so this is probably gonna fall in a second. But the fact that this piece of glass has been able to withstand the force of gravity, not only in keeping itself up, but keeping all these poker chips up, is pretty astounding. So there you go. Cohesion and adhesion. Now you'll never forget it. Okay, hydrogen bonds, super strong. All right, and you might do a lab with that. I don't know. Some teachers like to do labs with that and count pennies and stuff. It's a pretty neat one. So I'm gonna hop back in. Not that that wasn't a lovely little diversion. I'm gonna hop back into the PowerPoint. Any questions about cohesion and adhesion? So we have these strong bonds between water molecules. We have bonds between water molecules and other surfaces. And it allows things to walk on water. It allows water to travel up trees. Gives water a lot of great properties. Good? All right, I'm gonna move on then. So we'll talk a little bit about how water is the universal solvent. So this is really important as well. So water is capable of dissolving essentially everything except for oils, except for lipids, okay? And as you can see, this is kind of just a diagram of that happening. On the left, you can see that water actually kind of wraps around ions. So as I said, when you dissolve salt in water, what you're really doing is separating the Na positive and the Cl negative from one another, and you can see that water molecules actually wrap around those ions and kind of take them away from one another. So something to mention with water being the universal solvent is that those things that I'm talking about, like lipids, those are called hydrophobic molecules, things that are scared of water, right? And those are the only things that can't dissolve in water. And so anything that can dissolve in water, we call hydrophilic or water-loving. And that's going to help us majorly understand the plasma membrane when we get to talking about that. Because the lipids that make up the plasma membrane are not going to let water molecule things or anything that loves water or is similar to water or is ionic or has any sort of charge through the membrane because of this property, this universal solvent property, that it dissolves, water can dissolve anything except for oils and lipids. Okay? All right. Next one, love this one. It's a personal favorite. Ice floats, which sounds super, super simple. But one, if you think about it, it's pretty amazing because on the left here, you see some penguins floating under the ice. And obviously, a lot of bodies of water do ice over, at least on the top portion of the body of water during the winter time, right? And if ice sank, then anytime ice formed, it would just crush all the living organisms underneath it. We would have no marine ecosystems, right? So we have such diversity in life because ice floats at the top of the surface and therefore allows life to happen underneath without obviously crushing it or disrupting it, which is crazy and probably something that you don't think about often. But you can see on the right-hand side why ice floats. So this left picture shows that ice forms this, or water when it freezes, forms this really amazing crystalline pattern that actually makes it less dense than the liquid water, which is the diagram that you see on the right. Okay, and that's amazing because, again, over here you can see that we're actually less dense, which is why we're floating, than our liquid water. But this is the only solid that we know of that floats on its liquid, right? So any other solid that you can possibly think of that we could melt down to form a liquid, like gold, silver, like concrete, asphalt that the roads are made out of. If we had a liquid and a solid version of that and we plopped the solid into the liquid, it would sink. Every time when you're like, of course it would sink, solids sink, they're more dense. Yes, exactly, but water is not. And it's something that, again, you probably don't consciously think about every day because you just know it to be true. But it's a crazy property due to the fact that these hydrogen bonds form this really awesome crystalline pattern in ice that makes it less dense. Okay. All right, last thing that we'll talk about is specific heat capacity. So water is incredibly stubborn, meaning that it actually takes a lot of energy for water to warm up or to cool down. Okay, so water maintains, what that means is water maintains a relatively neutral temperature, regardless of like the outdoor air temperature. So as you can see down here on the bottom right, and I'm sure you've experienced this if you've ever been to the beach or anything like that. Like I'm obviously teach in Chicago, so I think about going to Lake Michigan. The sand in the summer is scalding. It's so hot, right? The sun is providing the same amount of energy to the water and to the sand, but the sand will increase in temperature incredibly rapidly, and the water will only increase just a tiny bit, even though it's the same sun that is shining above, okay? And that's all has to do with the specific heat of water. So the same amount of energy is going to have much less of an effect on the temperature change of water. Okay, so again, the same can be true in the winter, right? Water is not going to be 2 degrees when it's 2 degrees outside in Chicago, right? It's probably like 40 degrees in Lake Michigan. In the summer, when it's 100 degrees outside, the lake's probably 75 degrees, right? So it maintains kind of that middle range temperature, which is great for all of the cold-blooded or non-thermoregulators that live in the water, right? So they don't have to worry about maintaining crazy temperatures, really, really cold or really, really warm. And it's also just kind of a unique property. So you can see in the upper left-hand corner, I have a graph of the coast of California. And California is known to have like an incredibly moderate climate, doesn't get too hot, doesn't get too cold. It will really sit in the 70s, which is just perfect. Should definitely move to California. Pretty much all year, okay? So while that's maintaining its middle ground temperatures, if you go inland away from water, it gets really, 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 really hot, right? Like that's like 40 miles inland when you see, or maybe like 60 miles inland where you see like Palm Springs and San Bernardino, and they're getting incredibly warm. Whereas all of these cities along the coast are staying nice and temperate because they're really close to water, and water is maintaining that middle temperature. So that's a specific heat capacity. Questions about those 4 properties. So we have specific heat, ice floats, universal solvent, cohesion, and adhesion. Okay, cool. I'm going to go ahead and move on then, and we're going to talk about macromolecules. So now is where we get even to more of the biochemistry part as opposed to just chemistry stuff. So there's 4 macromolecules that we have that all life is made up of. And those are proteins, nucleic acids, lipids, and carbohydrates. You need to be relatively familiar with their general shape. You need to be relatively familiar with what atoms they make up and what properties they have. Okay, because all of those things are going to contribute to the way that cells are formed, the way that the cells act, they respond, cell signaling. When we talk about like, oh, this is a steroid hormone that's signaling, you need to know that that's part of the lipid family, and therefore it can go right through the plasma membrane, right? Like these things will circle back and have long-standing effects in the future. So it's important to get them down early. We're going to start talking about carbohydrates. So something to know, carbohydrates are made of carbon, hydrogen, and oxygen, and that is it. Okay, the monomers, so got to know the difference between a monomer and a polymer. I always use Legos to show this. So one Lego is a monomer, a Lego group is a polymer. It's a grouping of monomers, but the single subunit, if you broke this thing up into as teeny little tiny pieces as you could, would be the monomer. So the monomer here for carbohydrates is any type of monosaccharide like glucose or fructose. Glucose is probably the one that we think of most often because it's what we use as an energy source in cellular respiration. Okay, you can see glucose in the upper left-hand corner here. That's the structure of it in case you ever had to identify it. Usually that doesn't show up as like an identification on the AP exam, but you will have to know about glucose and what it would break up to. The formula there would be C6H12O6, just carbons, hydrogens, and oxygens. And then those things can come together to form polymers. So polymers, as I said, are groupings of monomers. We call them polysaccharides instead of monosaccharides, which should make sense. Examples would be starch and cellulose. These are really important to know about. You need to know that starch is how plants store energy. Because we get energy from glucose, we all store that differently. Plants store it as starch. If you've heard of like really starchy vegetables like potatoes, it just means they have a lot of sugar and a lot of carbohydrates in them because they store up all of that glucose in the form of starch, which is what you're looking at the bottom here. And you can tell it's just like pretty much looks like 6 glucose molecules just linked together there. Cellulose is another group of carbohydrates that comes together and forms structural material in plants. And then you can see maltose here is called something we call a disaccharide because it has 2 building blocks there. Something to look for: cellulose, glucose, fructose, maltose, all of those end in ose. When you see that, you know it's a sugar, and sugars are going to be carbohydrates. Okay. Examples of like food sources, things like bread, pasta. Most fruits are really high in carbohydrates because they have a lot of sugar in them. Any sugar is going to go into that family. All of those things would be really carbohydrate heavy. They give us the purpose of them. They give us really quick energy. But for the most part, it's like a sugar rush. It burns quickly and then we run out of it unless you have like a more complex carbohydrate like a whole wheat. Proteins are next. So now we're adding another atom into the picture. They're made of carbons, hydrogens, oxygens, and nitrogens. The monomer of a protein, the little building blocks that make them up, are amino acids. If you look over to the right here, you will see the 20 amino acids that make up all proteins on Earth. You'll notice something that I'll talk about in a second. They all look pretty similar except for one crazy side chain. And then the polymers are going to be something called a polypeptide, which you can kind of see down here as this string. It looks almost like a ball of or like a yarn. And then when that gets folded up, that's when we have our official protein. We have our 3D structure. Proteins need to be in a 3D structure in order to be functional. So on the left here, I'll spend a little time talking about this diagram. You can see it has 2 carbon molecules, but so basically that central carbon, and I was going to say this earlier, so I'll mention it now, that central carbon, let me plug my computer in really quick, is really, really important, and carbon is always going to be the most important atom for biology because it forms 4 really stable bonds. And you can see that there because you can see the 4 lines coming out of that central carbon right here. We have one connected to a hydrogen, one connected to something that we call a carboxyl group. So that's the other carbon connected to 2 oxygens and hydrogen, one with a double bond there. We have this R group, which is basically just like, you can think of it like a variable, like X. And that's where those crazy side chains come in. And then we have a nitrogen group. This is called an amine group, hence amino acid, and it has a nitrogen and 2 hydrogens. So we call it an amine. Okay, so central carbon, 1, 2, 3, 4 bonds off of it, 1 hydrogen, 1 carboxyl group, 1 amino group, and then we have this R side chain, which is just variables depending on what amino acid you are. So if you go and look back up in the upper right, you'll see all these crazy things hanging off. Each one of those is going to be the R group for that specific amino acid, which is what makes this guy a valine instead of a leucine, instead of an isoleucine, instead of a methionine, etc. Okay, so that R group is super, super important. Next, nucleic acids. I'm sure if you know anything about biochemistry, or if you know nothing about biochemistry, you at least know about nucleic acids because they are very, very commonly taught in every intro course. Here, and this is kind of cut off, but you do need to know this, they're made of carbon, hydrogen, oxygen, and phosphorus. Okay, so oftentimes on AP exams, they may ask you something about like a phosphorus containing macromolecule versus a nitrogen containing macromolecule. And you'll need to know that they're talking about a nucleic acid versus a protein, because nucleic acids have phosphorus in them and proteins have nitrogen in them. So it's just good to know the building blocks. The monomers of these guys are nucleotides. This isn't a great picture to show it, but the nucleotide in and of itself is a sugar. It's either ribose if it's RNA or deoxyribose if it's DNA. It's a phosphate group, which, as I said, this contains phosphate, and those phosphate groups are going to form this nice strong backbone here so that our DNA doesn't like cut in half. And then lastly, it's going to be the base that you see here. And if we're in DNA, it's going to be cytosine, guanine, adenine, thymine. If we are in RNA, it's all of those except we replace thymine with uracil. Okay? Something important to note about the actual biochemistry behind these bases is that, as you can see probably right off the bat, cytosine and thymine/uracil are really small. It's just one hexagon shape versus guanine and adenine, which have 2 larger shapes like a hexagon and a pentagon kind of glued together. Guanine and adenine are purines. Okay, that's just the category of nucleotide that they are. So I always think ag, like agriculture, is pure. Ag is pure. Adenine and guanine are purines. Okay, and you should hopefully remember from whatever intro bio course that you took that adenine always pairs with thymine or uracil, and guanine always pairs with cytosine. So it makes sense that those big, bulky purines would bind with either the cytosine, thymine, or uracil, which we call pyrimidines, which are much smaller. Okay? So if you think about it, if you have a big, bulky adenine on one side of your DNA strand, you want a smaller thymine on the other side so that we have nice, consistent, equal ladder rungs, right? If you picture DNA as a ladder. If we had like a thymine and a uracil paired, or a thymine and a cytosine, our ladder would be too skinny. And if we had a guanine and an adenine, it would get really, really fat. So we need to pair an adenine or a guanine, those purines, with a pyrimidine so that we have nice, even bonds. Okay. The bonds that form between base pairs, and this is really important to know as well, so like the bonds that are finding between the base pairs in DNA right here, are hydrogen bonds, which we talked about earlier, weak. Right? And that's really, really important that these bonds are weak because we need to break them all the time. Anytime we go to replicate DNA, step, replicate. Anytime we go to transcribe and turn this guy into an RNA messenger, step, right? So it's important that those bonds in between are hydrogen bonds because they allow us to break and to make those bonds much easier. Okay? Another reason why it's super important to know the biochemistry behind this, because it will make much more sense when we talk about other things Last macromolecule, the lipids, the fats. Oh, I should have talked about, so nucleic acids obviously are DNA and RNA. Proteins, great examples are all the meats. Nuts have a lot of protein in them. Eggs have a lot of protein. Milk has a lot of everything, but protein is one of them. So all of those would be naturally occurring things that have lots of protein. You need protein for your muscles to build up muscle strength. It's more of like a material thing. And the proteins are also the doers of the cells. So DNA actually does nothing, right? Like you're like, oh, the DNA is what makes me, and it is, but it only makes you you because it codes for a protein that actually makes you you. So the proteins are the unsung heroes of the cells. All right, I digressed, but we're back talking about lipids. Okay, so lipids are all the different fats. So you can think of like oil, butter, anything else. Nuts have a lot of fat in them. Avocados, which are all good fats in those guys. We're back to the basics, only made of carbon, hydrogen, and oxygen. The monomer of lipids are called fatty acids, okay? And they can really vary. And polymers, technically there isn't a polymer of lipids. It's kind of a funky macromolecule, but we have more advanced structures that come together, like triglycerides, phospholipids, and steroids. So all steroids are part of the lipid family. So again, we'll talk about this when we talk about cell signaling, but like our hormone or some like steroids are hormones, and those molecules, when they form signaling molecules, can actually travel right through the plasma membrane, which is also made of mainly lipids, which means that it can target any single cell in our body, which makes steroids really, 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 really helpful because they can travel anywhere. So a couple basic things here. So these fatty acids that I was talking about, we have saturated versus unsaturated fatty acids. I'm sure you've heard of saturated and unsaturated fats. So up at the top here, that's a saturated fat because every single carbon is saturated with bonds. They all make 4 bonds just like they should. And you can see that here going down the entire line, going down, 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 4 bonds all around. Unsaturated fatty acids, they don't have all their carbons saturated. So you'll notice that there's that bright red double bond forming between 2 carbons here, which puts a kink in the fatty acid. Okay, so saturated fats are all jam-packed with bonds and they lay nice and flat. Unsaturated fats have a V in them. So if you know anything about like health overall and what you should and shouldn't be eating or nutrition, things like that, you'll know that unsaturated fats are way better for you than saturated fats are. And hopefully this now makes sense because saturated fats can pack themselves into your arteries, right? Because they're nice and flat so they can stack up like bricks and they can clog up your heart much more easily or your arteries or anything like that than an unsaturated fat, which is kinked and therefore can kind of bounce freely throughout your bloodstream and it's not going to stack up like a saturated fat would. So it allows for better traveling through the blood vessels and therefore won't create as many problems for you in that capacity. Other things, so I do want to show you this phospholipid down here because we're going to talk about it extensively when we talk about the plasma membrane, which we could talk for hours just about the structure of the plasma membrane if you guys are interested. But phospholipids are really crazy because they have a phosphate group, hence phospholipid. You can see that here, there's a little phosphate right there that forms this head region, and all of these things are polar. Okay, so phosphate groups are going to be slightly polar. And so remember when I talk about polar, I talk about uneven sharing of electrons. It's also similar to a water molecule, which is very polar. Okay, and those things are going to be on the outside of the plasma membrane, as opposed to these fatty acid tails, which you can see have a saturated and an unsaturated, a saturated and unsaturated fat in them, which are obviously going to be very, very non-polar because they're made of lipids, and lipids are very hydrophobic. If you've ever put oil and water together, you know they don't mix. Okay, so with the cell membranes, it's a dual layer here, which you can see down at the bottom where we have the polar heads on either side with all the fatty acid tails sticking on the inside. And our cells, just like the rest of our body, are full of water inside and outside. So it should make sense that those polar heads are lining up on the outside of the membrane because those are the ones that are interacting with the water molecules, right? Water inside, water outside. Polar heads are kind of like the barriers of that water on either side of the plasma membrane. And then all the fatty acid non-polar tails are going to be hidden in the inside. I always think of them like octopus like tentacles that are going to try to grab anything that is water loving or like water and not let it through. Okay, so these principles are pretty important just in getting an understanding of what can and cannot get through the cell membrane. So if anyone has any questions, that's actually all I have. Biochemistry, while it can be a little bit complex, the basics are what I just talked about. So knowing the 4 macromolecules is really important. We're going to talk about the origin of life on Earth next week. And so you need to know how lipids, carbohydrates, amino acids, and DNA, RNA behave in order to see how they spontaneously come together. You also need to know about water for sure, and you need to know about those bonds and those electrons. So if you have any questions, I can stay on for the next 4 or 5 minutes, or as long as we need, I suppose. And I'd be more than happy to answer them if you have specific things that were confusing to you. Otherwise, definitely tune in. I want to really, really briefly mention a couple things about what Fiveable is going to look like for you guys. So basically what you can do if you're interested is you can subscribe to my subject area or multiple subject areas, and what that will give you access to is all the live sessions, but even more importantly, it will give you access to the replays of the sessions that we're talking about. So why would that maybe be important? Well, let's say it's May and you have a really large AP biology exam coming up and you really want to know what it is that you totally forgot about biochemistry or any number of other things, right? So you can replay any video, you can fast forward through it and just get the information that you need. So what you can do, so at the end of September, so our live sessions were free through September, but September is almost over. Time is flying. So Monday is the start of October. So if you want to keep access to the replays and watch any of my upcoming sessions, you'll actually need to join Fiveable. So until October 1st, if you sign up before Monday, so if you sign up tonight, it would be awesome, or tomorrow or this weekend, you only have to pay $15 per subject, and that gives you access to all of my live sessions for the rest of the year, as well as replaying anything. $15 to do all of that, okay? Which is amazing. That's like, I don't know, a large meal at McDonald's. You can also pay $30 for all subjects all year. Okay, so that way, if you're in multiple APs, you can pay $30. If you're in 3 APs, that's a steal. And you can watch any videos as well as watch any replays when it's May and you're confused. It's a one-time payment, so it's way cheaper than any other live support you can find. And last year, Fiveable actually saved students over $1 million in college credits because kids got credit from taking their AP exams. So therefore, they were able to opt out of those credits in college, and college is the most expensive thing of all time. I'm still paying off college, so definitely worth it. And there should be a link that I can send out that you guys can sign up therefore and get that early bird special. Okay, cool. So pretty straightforward. And hopefully that makes sense. But again, so $15 will get you access to my content. Okay. All right. That's all I have for you. Please sign up. Please sign it. And I'll be on the line for about 5 more minutes. I know Charisma has a question. I would be really, really happy to answer for her. If anyone else has a question, feel free to stay on. Otherwise, have a great night. Be safe, and I'll see you next week. All right. So Charisma. Oh, Joshua, you're so welcome. It's one of my favorite demos. Love doing demos. Makes live sessions more interesting. It's really hard to like not get to see you guys and interact with you and just be like talking. I don't like hearing myself talk this much. So I'll try to bring in fun and exciting things when I can. Okay, so noticeable differences in the structure of each macromolecule that can help identify which is which. Would love to go through that because it's really, really important to know. So I'm going to share my PowerPoint with you again, or share my screen of the PowerPoint with you again so you can follow along. And I'll point out a few key differences. So lipids are actually the easiest. So we can start there, which is what my screen is on already. Because they're super, super unique in that they always are going to have that long hydrocarbon tail is what we call it. So lots of hydrogens connected to lots of lots of carbons connected to lots of hydrogens in a long chain makes those tails like you see there. And those like long strings of carbons and hydrogens, carbons and hydrogens, carbons and hydrogens. And that's all it is key lipid right away. Nothing else does that. Everything else would have oxygens mixed in, or it would form like a hexagon or a pentagon or some sort of shape. So if you just see long chains of carbons and hydrogens, immediately, easy point, that thing is a lipid. Okay? I should have checked that I was answering the question. So that thing is a lipid if it has that long carbon hydrogen tail sticking off the back, and that's what you see. It sometimes will have like a funky head group or something like that, but the tail is a giveaway. Nucleic acids, so things to look for. So usually you'll see nucleic acids are three-parters, which I don't have a great picture of. I really should add one in there. But they're going to have like a sugar group. They're going to have, I'll find one for you guys really quick. Sure. Send you guys a link. Here we go. Oh my gosh, that is the longest URL I have ever seen. It won't even let me send it. So I will just embed it into the PowerPoint slide instead. Sorry. Here we go. Come on, why is everything being slow? Okay. Ta-da! This will be helpful. So it's this three unit grouping if you're looking for nucleic acid. So that sugar, it's always going to be a pentagon. Both ribose and deoxyribose are going to be pentagons. You'll also see some sort of hexagon or hexagon-pentagon situation coming off the top, which is going to be your actual base. And then you'll see a phosphate group. If you see something that has those three distinct units like that, that's going to be a nucleic acid. You'll also see nitrogen and phosphorus in it. So nucleic acids do have nitrogen. You'll see all of our bases have nitrogen in them, but they also have phosphorus. So if you see something that has both nitrogen and phosphorus, that's going to be a dead giveaway that it's a nucleic acid as well. Okay, so that's your differentiator for that. For proteins, here you're just going to look for the nitrogen and the carboxyl group and then some sort of side chain. So by carboxyl group, it will either look like C double bonded to an O bonded to an OH, or you'll see over here, if you're looking at the amino acids, they just say COOH. If you see basically a four-pronged group, one has COOH, one has a nitrogen-hydrogen pair group, then you know that you have that amino acid because they'll always have four prongs, right? A hydrogen prong, a carboxyl prong, COOH, right? An amine prong, which is NH2, and then some sort of random fourth. Okay, so that's your giveaway for proteins. They will definitely have nitrogen in them, and they'll also have that carboxyl group, COOH. Lastly, carbohydrates are pretty much the easiest. They're always going to be hexagons, and they always have carbons, hydrogens, and oxygens, and that's it. So if for some reason they like pull a nucleotide and they have a hexagon over here and a hexagon over there, you can just look and try to find the one that just has carbons, hydrogens, and oxygens in it, because that's all carbohydrates are. But they form that nice hexagon shape as opposed to the carbons, hydrogens, and oxygens that form our fatty acid tails in our lipids. Okay, so hopefully that helps answer your question, Charisma. If you look at pictures enough, they all share those common characteristics like that, and you'll get used to seeing them. But yeah, those are the basics. Okay. All right. Anything else that I can answer for people? I'm trying to delete this entirely long URL for that one silly little picture. Any other questions? Silver, so glad it was helpful. I will try to make things as helpful and relatable. I've looked at a bajillion and 5 AP exams. Yes, so Joshua, to sign up for the future, let me get you that link right now because that's super, super important. So I will send you the Fiveable website link, and on that link, you can just sign up to here we go, learn more and sign up to, why is there so many buttons to click? Gosh, here we go. You can sign up. Again, it's one payment of $15 as long as you sign up before Monday. That's crazy cheap for getting live sessions every single week and replays whenever you want, including, you know, exclusive content, videos. I'll have worksheets on occasion. You can obviously ask questions to me live. So definitely sign up because it makes things more interesting when there's people who are watching and listening to me and it's not just me talking to myself. Okay, the link's there for you. It was great working with you all tonight. Again, have a great night, be safe, and I'll see you next week to talk about origins of life on Earth. Talk about how crazy that is, that life started out of nothing. Stay tuned. See y'all then. Bye.